So in the previous two slides, we discussed the basic principles. Now, if you're a small organism, that's fine and good, and you can probably run everything inside the few cells you might be possessing and then get rid of urea. Humans are much bigger and more complex, and they actually do go through an additional step of nitrogen management. So in all the other tissues, there are plenty of glutamine transferases that will take glutamate, and then that will take their own alpha ketoglutarate and collect the amino groups onto glutamate that they converted from alpha ketoglutarate. Now, the next step would be to dump glutamate into blood, except glutamate is obviously an acid. It's glutamic acid. So if you dump too much glutamic acid into the blood, the pH of the blood changes, and as we hopefully remember, that comes with all kinds of problems. We don't want to have acidic blood. Even a small change in pH would be deleterious. So instead, other tissues in our body repackage the glutamate as glutamine by attaching an additional amino group here using glutamine synthetase that uses ATP, that's why it's synthetase, to make glutamine, which is uncharged. It's polar, but it doesn't inherently carry a negative charge because the carboxyl group has been balanced out by the amino group. So L-glutamine is then secreted into the blood and is taken up by the liver, which will use a glutaminase to convert it back to glutamate or glutamic acid, which then will undergo the glutamate dehydrogenase reaction and inside the mitochondria of the liver and release the ammonium ion for the urea cycle. Now, as you can see here, that I primarily talk about liver, but our kidneys and our intestines can also participate to a much lesser level in the collection of the ammonia out of the blood if liver is not fulfilling its function. But mostly the ammonia is collected in the liver. Now, since an additional wrinkle in the story is that while most of our tissues use glutamine and asparagine in order to get rid of their own personal excess nitrogen and put it into the blood, muscle actually has an additional problem. It, never mind nitrogen, it tends to overaccumulate pyruvate when it's running a lot of glycolysis reactions and runs out of oxygen. So when there's too much pyruvate in the muscle and the mitochondria of the muscle cannot keep up with the amount of pyruvate produced in the cytoplasm, in order to keep the glycolysis reaction going, the muscle then transaminates pyruvate to alanine, because that's what basically alanine is, a pyruvate with an amino group attached. Using the same glutamate to alpha ketoglutarate reaction, it takes the amino group from glutamate, which in turn it got it from the various amino acids the muscle happens to have, coming from the proteins of the muscle, of which muscle has a lot because it has all the sarcomere machinery going on. And the alanine aminotransferase converts pyruvate to alanine. Alanine is then dumped into the blood, and glycolysis can keep on going because the pyruvate is being removed from the muscle as alanine that goes into blood then. And now, not to waste a good thing, your liver will take up the excess alanine and convert it back to pyruvate, right around gluconeogenesis, make more glucose, and give the muscle more glucose to work with. So this is the glucose-alanine cycle, which runs together with the Cori cycle, which, if I'd like to remind you guys, is the lactate into the blood and then gluconeogenesis into the liver cycle. So this, what I just told you guys in all of these slides, This is summarized in this nice diagram here I found online. Here's the typical transamination of that all cells undergo when they have too much of one or many amino acids. They will undergo transamination, collect it all onto glutamate, and then convert it to glutamine in order to put it into the liver, put into the blood and into the liver. And muscle will collect some of it. It will run this reaction as well, but if it has specifically pyruvate accumulating, it will package it as alanine and send it to the liver as well. And it is liver overwhelmingly that runs the urea cycle. Kidney and our intestine can help with running the transamination of various transamination reactions, but it's the liver that runs the urea cycle. So this is what the slide here is showing. Now, the urea cycle is a process that can take two nitrogens, combine them together, and make urea, which is, you can see right here. And the two nitrogens come from different locations. The first one is the ammonia nitrogen that the ammonium ion that we just talked about, the one that will come off the glutamate that the liver has collected using the glutamate dehydrogenase. And then the second nitrogen comes from the aspartate shuttle that we'll discuss a little bit later, that is replenished via the TCA cycle. So the urea cycle and the TCA cycle are actually intrinsically linked in the liver, inside the liver. And unlike the TCA cycle, it completely occurs in the mitochondrial matrix. The urea cycle actually is separated. Part of it occurs in the mitochondrial matrix, where the ammonia ion is bound up with carbon dioxide into the reactive carbamoyl phosphate, which is then fed into the cycle. However, most of it actually occurs in the cytoplasm, so all the arginosuccinate, arginine, and ornithine conversions all occur in the cytoplasm, as well as urea is produced in the cytoplasm of the liver cells. Once urea has been produced, it will be received in blood. It will be released into the blood again and collected by our kidneys and excreted in urine.